Hello everyone. In this screencast, we're going to be discussing how you're going to be harvesting the bacteria in this second week of this 4 weeks long experiment where you started last week growing the bacteria here. This week we're going to be harvesting them, as I say. Next week we'll be extracting the genomic DNA, and then finally we'll be quantifying the DNA that you have extracted. So we're on step 2. You learn how to harvest the bacteria. So in order to do that, what you'll be doing is first you'll be preparing a buffer that then you'll be using to scrape the bacteria out of your plate. And then finally you'll be concentrating the bacteria into a smaller volume by centrifuging them for 10 minutes at the very end of the experiment. So now your bacteria have been growing overnight at 37 degrees on the plates of media that you made. So now you'll be starting basically with these plates filled up with a nice layer of bacteria, each one of them, if everything went well. And this bacteria obviously should be E. coli, the E. coli that you added on that, and hopefully not contaminants. So now you'll be giving these plates, and then you have to scrape this bacteria out with a buffer. So first thing, you'll be preparing the buffer. The buffer, in general, the definition of a buffer will be a solution that keeps the pH stable. However, when you work in a lab, buffer has a more of if you want to extend the definition, where by buffer we often mean a solution that optimizes whatever process you're trying to do. For example, if you're using a bacteria, a buffer will be a solution that optimizes the activity of the enzyme you're trying to use, which would do it by stabilizing the pH, but not only, sometimes even by containing some salts, which maximize as well the activity of the enzyme itself. So in our case, the buffer that we'll use is a solution that optimizes what you're trying to do, which is harvesting the bacteria, waking them up, and then getting them ready to lyse them and extract the DNA. And so in order to achieve that, what you have to do is prepare a solution which contains these three components, which are tris, that's the buffer that keeps the pH stable. Then we have a molecule, which is a chelator for cations, divalent cations, which is EDTA. And then we have sodium chloride, regular salt. And this solution has to be at pH 8. So why do you need it, first of all, before we discuss how you make it? Well, the reason why you need it is to optimize the process that you're trying to do. So now, if you look at a bacteria, you know they have a cell wall and a cell membrane right here. The cell wall, and therefore the cell membrane stability too, is dependent particularly on the presence of, it's made by peptidoglycans, and it's dependent on the presence of different ions. Among them, calcium is extremely important, and magnesium as well, but particularly calcium. So now, if you add EDTA, which is part of your buffer, now what you're doing is basically chelating, or basically removing divalent ions like magnesium and calcium. And so by removing the calcium, you're actually weakening up the structure of the cell wall. So this cell wall is becoming less strong and easier to attack next week when you use the enzyme lysozyme. However, this has an effect. I don't know if you remember from Bio 1, you should have learned that one of the reasons why bacteria need a cell wall is because their cell membrane is weaker than the eukaryotic cell membrane. And the reason being that their cell membrane does not contain cholesterol. So now you're weakening the cell wall, so even the cell membrane will be weaker. So there is a chance that now, as soon as you collect your bacteria can actually explode and release all of their contents. You don't want that to happen yet. Okay, right now you're only harvesting your bacteria, so you want to weaken the cell up without them blowing up. And you can avoid that by basically creating an isotonic solution, which you do by adding sodium chloride. So the reason why you add the sodium chloride, the desired concentration, is to make sure that the pressure on the cell membrane of the bacteria is not too strong, so that bacteria will not explode. Then another reason why you add the EDTA is because once you actually blow up the bacteria and you release the DNA, the bacteria contains also some enzymes, like we all do, called nucleases. These are the enzymes that basically cut the DNA, they destroy the DNA. You don't want that to happen. You want to extract structurally integrate DNA, perfectly sound DNA. And so you want to inhibit the activity of these enzymes, and then the way you do it is that by blocking basically magnesium, which is extremely necessary for these enzymes to perform their actions on the DNA. So if you remove the magnesium with the EDTA, you actually block their activity. Also, the pH itself, pH 8, is non-optimal for these enzymes. So if you bring the pH at 8 by adding tris HCl, as we are doing, you actually minimize the chance of this enzyme acting on the DNA. So that's the reason why you add all of these components, so EDTA, sodium chloride, and the buffer, and you bring a pH 8. So how do you prepare this? Very simple. You're basically preparing just a solution with a tiny little difference at the end, which is that you have to adjust the pH. So you weight the amount necessary of tris, EDTA, and sodium chloride to achieve the concentration that you want. Okay, like when you make any solution. Please keep in mind that when you do this calculation, you have to take into account the formula weight of these molecules, which is written in the bottle that you find in the lab, not the theoretical one. The reason being that some of these molecules may come hydrated, meaning they can contain additional molecules of water, which would affect the final formula weight. So again, you have to do your calculation based on the formula weight you find on the bottle in the lab. So once you have weighted them, you add them all together in a 250 milliliter beaker. You have to prepare 100 milliliter solution of buffer, but you'll first resuspend these molecules in 70-80 milliliters of water. The reason why you do not go straight for 100 milliliters is because then you need to add additional volume of 1 molar HCl to adjust the pH of this solution to 8, the pH you want. So you have a few milliliters of HCl. You'll do this by monitoring the pH with a pH meter. Okay. You add the pH, you reach the pH 8. Once it's reached, then you transfer everything into a 100 milliliter volumetric flask, and then you bring the solution to the final 100 milliliters total. You mix it, your buffer is ready to be used. Now, a few words about the pH meter. First of all, I will not help you on calibration and how to use it. You're supposed to know how to use it. However, if you have any doubt, go and watch the video that is on Blackboard through Senetex. So you go through Senetex, you log in with your own password, you select how to use a Spec 20, all the information is there. And actually it's relevant because the pH meter is used there is exactly the one that you will be using. So review that. Now, other things I want to remind you is that when you work with a pH meter, you're always very gentle with an electrode. They're very delicate, made of glass, very easy to break. And also, to avoid cross-contamination of your buffer, or also the solution, the standard solutions that you'll be using, you always rinse the electrode whenever you transfer it from one solution to the other. Okay, just rinse it a little bit of water. And then another thing that I want to suggest to you, just to make it easier, and based on what I see from other students, when you are using the pH meter, you put your flask here, so your beaker, and then you keep mixing the solution with a stirrer. So this way you don't have to use one of your hands to constantly stir the solution. Keep in mind, however, that the stirrer, first of all, when you use this type of plates, you don't have to turn on the heat. This would affect the pH. And make sure that the electrodes is not hit by the steering bar, okay, which obviously could break it. Okay, so now you made your buffer, you're ready to harvest your bacteria. So now you'll take your 5 plates, and in each plate you'll be adding 2 milliliters of this buffer using a pipette. So you add the 2 milliliters, let it soak a little bit, and then using the hockey stick, the same stick that you used last week to play the bacteria, now you will actually scrape the bacteria out. You'll do the same movement that you did last week. At this point, you do not need to work in a semi-sterile environment. Your bacteria, even if you contaminate the plates, the contaminant bacteria will not have time to grow. So you can actually work on your regular bench, still clean it with some isopropyl, but then you can just open them, even if you're far from without the Bunsen burner, basically. So you scrape your bacteria out, you be gentle. You want to get only the bacteria which are on the top of your media, but not the media. You don't want to scrape the media out, okay? So gently, you remove them, get them out, and then you collect the 2 milliliters plus the bacteria into a centrifuge tube. Once you have done that, you do a second round, so you rinse each plate with 2 more milliliters of buffer. You want to make sure you collect as much bacteria as possible, and then you transfer again all of the bacteria from the second wash into the centrifuge tube. So in total you should have about 22 milliliters considering the volume of the bacteria in here, okay, inside this tube. Great, so now you have your bacteria in 22 milliliters, whatever it is, and now you want to concentrate them, and you'll do that into a smaller volume. So you'll do that by taking your centrifuge tube, you'll match it on a top loading balance with the tube from another group, and then you centrifuge it for 10 minutes at a speed which you can determine based on what's in the lab manual. So review the lab manual there. Once you spin it for 10 minutes, you can discard the supernatant. The supernatant will contain the old buffer that you use plus some contaminant media, and the pellet should contain your bacteria. So your bacteria now can be resuspended in 5 milliliters. So as you can see, you're concentrating them from 22 milliliters to about 5 milliliters of buffer. You resuspend them well. Make sure that all the bacteria are resuspended, that you don't see a pellet anymore. And once you're done with that, you transfer them into a test tube, clean test tube, and you store them. You put some parafilm and we'll store them at minus 20 degrees. So we'll freeze them until next week when you're ready to actually destroy this bacteria and collect the DNA out of it. Together with this test tube, I expect you to give me 2 more test tubes, clean test tube filled with buffer. Okay, we'll need your buffer for the following week as well. So 2 test tubes will be plenty. So you fill 2 test tubes, put some paraffin, and you give them to me. Don't forget to initial so you know what is yours, and obviously don't forget to label as well your bacteria. Okay, that's pretty much everything we had to discuss for this week. In terms of safety, the same concerns that we had last week minus the Bunsen burner. So you're still working with bacteria, potentially harmful to you. So everything we discussed the week before still applies in regards to the bacteria. Okay, see you all in class. All right, take care.